Hello guys, we are from group 16. Today, we are going to explain about protein denaturation. So, what is mean by protein denaturation? Denaturation disrupts the normal alpha helix and beta sheets in a protein, and uncoils it into a random shape. Denaturation of proteins involves the disruption and possible destruction of both the secondary and tertiary structures. Since denaturation reactions are not strong enough to break the peptide bonds, the primary structure, sequence of amino acids, remains the same after a denaturation process. Now, let's see how this mechanism of protein denaturation works. Unfolding of native proteins occurs at both lower and high temperature. At higher temperature, it is called heat denaturation, or thermal denaturation. Denaturation that occurs at lower temperature are called cold denaturation. During the protein denaturation, it will breakage of hydrogen bonds, disulfate bonds, hydrophobic interactions, and van der Waals forces. But there is no breakage of peptide bonds. The heat will disrupt hydrogen bonds, and nonpolar hydrophobic interactions. The kinetic energy is increased due to the high temperature. Cooking will cause vibrate to the molecules. The amount of heat determines the amount of motion. More heat, more motion. Enough heat applied to the molecular motion will cause the bonds to break. The protein denature returning to their primary chain-like structure, result in formation of a solid gel. The gel entrapped water molecules inside the white into a semi-solid structure which holds its shape under normal conditions. Okay. Next is agent causing denaturation. There are two categories of the agents that can cause protein denaturation, which is physical and chemical. The first one is heat. Most protein can be denatured by heat, which affects the weak interactions in a protein, which primarily hydrogen bonds in a complex manner. The second is hydrostatic pressure. Protein undergo dissociation and unfolding by pressure, mostly because the final states are more hydrated, fewer non-hydrated cavities and occupy smaller volumes. The last one is UV radiation. UV radiation supplies kinetic energy to protein molecules, causing their atoms to vibrate rapidly and disrupting the hydrogen bonding and the dispersion forces of protein molecules. The second agent of protein denaturation is chemical agents. First, acid and alkali. Acid and bases disrupting salt bridges, held together by ionic charges. Organic solvents such as ether, alcohol, acetone can also be the agents of protein denaturation. All of these can disrupt the hydrogen bond between protein molecules, and new hydrogen bond are formed instead, between the new alcohol molecule and the protein side chains. Lastly, detergents are amphipathic in nature, which having both a hydrophobic and a hydrophilic side. Proteins have hydrophobic and hydrophilic sides, meaning that the detergent will be attracted to these and forces the protein apart. Okay, let's see how protein denaturation occur at each level of protein. In primary structure, the sequence of amino acids held together by covalent peptide bonds, which are not disrupted by the process of denaturation. In secondary structure, proteins lose all regular repeating units or patterns such as alpha helices and beta pleated sheets, and adopt a random coil configuration. There are two structures which are alpha helix and beta pleated sheet. In tertiary structure, involves the disruption of the following bonds. Firstly, covalent interactions between amino acid side chains such as disulfide bridges between cysteine groups. Lastly, van der Waals, which known as induced dipole interactions between nonpolar amino acid side chains. In quaternary structure, the protein subunits are dissociated, and the spatial arrangement of protein subunit is disrupted. Thermodynamic of protein denaturation. Denaturation is a phenomenon that involves transformation of folded structure of a protein, formed under physiological conditions, to an unfolded state under non-physiological conditions. The conformational changes in proteins invariably affect its chemical and physical properties. This protein denaturation can be studied by monitoring changes in the physical and chemical properties. So, this is the graph of thermodynamic of denaturation. Yn and Yd represent the values for the native and denatured states, respectively. As denaturant concentration increased, the value of Y remains unchanged initially, and above a critical point its value changes abruptly from Yn to Yd within a narrow range of denaturant concentration. Once a protein molecule begins to unfold, or once a few interactions in the protein are broken, the whole molecule completely unfolds with a further slight increase in denaturant concentration. Now, let's see the advantages and disadvantages of protein denaturation. Advantages. Stomach acids denature protein into simple amino acid chain that digestive enzyme can easily break it down. It is a large-scale study of protein. It is important to study protein as represents the actual functional molecules in cell, such as cheese, paneer, and coa. Knowledge of concentration of protein will help to determine the interaction between sugar and amino group in Maillard browning. Disadvantages. It's due to the loss of both secondary and tertiary structures. Enzymes will lose its shape. The changes in active site will reduce the effectiveness of enzyme. As a conclusion, the process of unfolding of protein is referred as denaturation, thus causing disorganization of the internal structure of protein. Denaturation affects secondary, tertiary, and quaternary structures, but not the primary structure, peptide bond. In addition, the process of denaturation may be reversible or irreversible. It is based upon the nature of amino acid residues which participates in its structure. So, I guess that's all for our presentation about protein denaturation. I hope you guys can get something in this video. Thank you. Thank you for watching. Bye bye.